that time we had the possibility to decide for another expression of this protein in lung tumors that are adenocarcinomas. And as we said before, the first finding that this protein is immunogenic in humans as evidenced by serum antibodies. Of course, I mean, we tried to have from cancer cells in culture to have the possibility to identify the same protein, and in this case we have like a monomer around 50,000 kilodaltons, and at that time we used some immunoperoxidase technique for pathology, and with the first epitope we were able to show the activity concerning some lung carcinoma, and you'll see with especially epithelial carcinoma, adenocarcinoma in a major magnifying, and we have some positivity for some colon carcinoma, the same cystomatic origin as lung, and the very early stage of interstitial fibrosis, and no activity in breast carcinoma, no in urothelial carcinoma. And since our studies showed that there was some similarity, maybe one or two amino acids less of the choline protein, we used against the variant the production of some antibody for A and B variants, and we have just a slightly positivity for myocardium. In another lab in San Francisco Tumor Institute, Bill Hughes was able to show by macroarray that there was a very interesting profile concerning especially the possibility to have a good specificity, but especially it was also a nice reactivity concerning the non-small cell lung cancer. And finally, the work was carried out for some at Tor Vergata University of Rome concerning some immunofluorescence studies, and of course, I mean, concerning especially the colorectal cancer, and was done some experimental model concerning that model, looking at liver metastasis and subcutaneous tumor, and the mechanism of action by cytotoxic activity of different cells. And here we can see the data that these people were able to reach concerning especially the rats that were vaccinated by the antigen, and the possibility to really to see a difference in the number of the metastases in the liver, and especially the nodules in the peritoneus. Of course, I mean, the cytotoxicity about the non-artificial resistant cells was good in comparison with control, especially according to the concentration of the antigen that was injected, and very significant data, and also concerning the other sensitive young cells, concerning again also the difference in concentration. At this point, we can say that there are also different mechanisms that we today know about tumor growth, tumor necrosis, and so it's possible that the cancer cells, we have also some special immune cells in the stroma. We can block the blood to the tumor. As you know, the activated T cells are able to produce interferon gamma and tumor necrosis factor, and so we have a good